Hi, I'm Lora Hambrick, and I'm going to be talking about the biochemical hallmarks of apoptosis. So, for the next couple of slides, we're going to talk about the DNA fragmentation and how it happens. So, a DNA coils around the nucleosomes, and that only involves uh, the nucleosomes at 180 BP. This causes the DNA between those nucleosomes to be connected by a linker DNA. When this linker DNA is cleaved, it's only cleaved during the DNA fragmentation, and that fragmentation only happens about around 200 BPs usually. So when you're looking at this figure on the right, where marker where the numbers 2 and 3 are, that's about 200 BP. If oxygen in the cell is absent, then the cell becomes anaerobic. This allows nitric oxide to induce the apoptosis and causes the cytochrome C to be released from the mitochondria. The mitochondria also releases a second mitochondrial activator of capases into the cytosol. Capases are proteins that are cysteine dependent and aspirated specific proteases. So to continue, uh, for apoptotic DNA fragmentation, the enzyme is responsible for its capases activated DNase. This enzyme is usually inhibited by the ICAD, which is the inhibited capases activated DNase. But during apoptosis, the ICAD is cleaved, and because that's cleaved, it allows the capases activated DNase, the CAD, to be activated. The CAD is what cleaves the DNA in the linker DNA, which is causing this DNA fragmentation, and why it produces the DNA fragments at about 200 BPs. Next, I'm going to talk about two more biochemical hallmarks of apoptosis. So the first marker is nuclear condensation. This usually happens during the apoptotic execution, and chromatin undergoes a phase change from a heterozygous, usually genetically active network to an inert, highly condensed form that is fragmented and packaged into apoptotic bodies. So I found a paper that talked about nuclear condensation, and what they're trying to do is figure out if this has stages or not. And to do that, they used a cell-free system to examine the roles of the capases and other proteases in the apoptotic chromatin condensation and nuclear disassembly. So this demonstrated that the population of the nuclei undergoing apoptosis in vitro appears to follow a reproducible program of nuclear condensation, and this suggests the existence of an ordered biochemical pathway. So while they did all of this, it was shown that there are 3 stages to nuclear condensation, and that would be a ring condensation, necklace condensation, and a nuclear collapse disassembly. So the ring condensation has to do with the nuclei collapsing. Then the necklace condensation has to do more with the shrinking of the necklace into compact aggregates. And stage 3, which is the nuclear collapse or disassembly, has to do more with the ATP is really prevalent here, and that just shows the collapse and disassembly of the cell. So the last marker I'm going to talk about is the phagocytosis of another cell. And the article that I found talked about how apoptotic cells of biphagocytic cells have a significant role in inflammation of dying cells. So apoptosis induces cell surface changes that are important for the recognition and engulfment of cells by phagocytes. The phagocytic removal of apoptotic cells does not elicit the pro-inflammatory response, but apoptotic cells engulf appears to activate signals that suppress release of the pro-inflammatory cytokines. So this figure is basically showing that it has several receptors that are implicated in the uptake of apoptotic cells by phagocytes. These receptors interact with the ligands of apoptotic cells either directly or by bridging the proteins. And the mechanisms that are involved in the LX recognition of apoptotic cells are indicated in red that you can see in this figure here.